That's blackmail material. And now the recording is starting with me saying blackmail material. Great. Uh, that's all of these recorded lectures. Anyway, it is October 6th. This is ESM 105. And we're going to get back into our week 5 through 6 material. I just got the 7 through 8 PowerPoint all polished up. I think it's looking good. We're going to start on that. I wanted to remind everybody, wait, this is Bio 105. That's the wrong, you want to do Bio 105 lecture? Let's go over here. I wanted to remind everybody, I posted an announcement, okay? But I did record this weekend a little tutorial video about here's how you could go about doing your annotated bibliography. If you need some help getting started, this is how I would approach an assignment like this. Just have a broad concept and then start Google Scholaring and start collecting citations. And then once you've got your 20 citations, you can go back and write all your summaries of each citation. It could work. There's a video, I think it's 20 minutes because I ramble. You get to see me on the weekend. There's my little fish tank. Isn't that cute? All right. So, yeah, that's up there. Don't forget that it exists. Okay. If you get started now, life will be way better. Remember, it's only worth 50 points in total, but you have to turn it in to pass the class. Okay? So make sure you do the dang thing. Okay. Any further questions? Yes, please.
Does that mean it's weighted a lot?
It's not weighted a lot. It's just that if you don't turn anything in, I won't input your final grade.
Hypothetically, if you did the world's most mediocre job possible, it wouldn't bring your grade down.
Yeah, I mean, it would because you would get less than 50 points, but it wouldn't necessarily dump you down to an F, Yeah.
That's just the hypothetical.
Absolutely. No, if you just sent me the 20 citations with a sentence after each, I'd be okay, well, you're probably getting 10 or 15 out of 50, but you won't automatically not pass the course. Solid?
Yes.
Question, yes. Oh, that tank just has a bunch of, what are they, mollies. My partner does all the fish tank management. It's got mollies and tetras in it. But then we've got water frogs in the other tank, and then a beta in the third tank, a female beta. And then we just got a giant millipede, which is the new exciting thing. It's super cool. Yeah. It's awesome. I didn't think I would like it. I thought it'd just be a creepy thing, but it's great. Cool. Oh, quiz 3. That's the thing to talk about. Quiz 3 is coming up. It's going to be covering the week 5 through 6 PowerPoint. It is due October 12th. Cool? We're doing great. Everything's going great. Okay. Ready? Here we go. Population growth models. All right. We talked a little bit about the geometric and the exponential growth models. I wouldn't say I'm necessarily proud of the quality of explanation I gave you on these two models here, but what I will say, okay, is that I'm really trying to just lead you into this model, okay, the logistic model of population growth. Because the logistic model of population growth it actually sort of approximates reality a lot better than the geometric or exponential growth rates do. Geometric works on very short time scales, and exponential works with specific types of organisms. Like bacterial growth, exponential's pretty accurate as long as you keep supplying them with media, okay? But the logistic model of population growth incorporates a carrying capacity, which is determined primarily by intra-specific competition. And why do I bring this back? Because I'm going to bring back the discussion of R versus K selected species, if you remember that from week 3 through 4 PowerPoint. All right? So you don't have to memorize this equation. I'm not even asking you to do calculations with this equation, but you're gonna see it again, rest assured, and I want you to understand what it's modeling, what it's meant to show. Cool? All right, so we've got this R value, we've got this K value. The R value is the maximum, it's called the intrinsic growth rate for a population, okay? Or the intrinsic rate of increase. It's the max rate that a population can reproduce if there's nothing constraining the population's growth. The K value is for carrying capacity. K stands for carrying. All right, have I done that joke yet? Either way, okay, cool. Well, it didn't work, so we'll just assume it's because I did it before. Carrying capacity, max intrinsic growth rate. Questions about those terms? All right, this whole equation just shows that, oh, as the population reaches the carrying capacity, it's going to level off and growth will slow down and stop. Okay. Good. So, density-dependent and density-independent population regulation. It is in every ecology textbook. They always talk about this concept. Just on the whole, like 5 pages talking about it. At its core, it's pretty simple. All right? Density-dependent, these are the 2 ways that population growth is slowed or that a population's size is regulated. A population's size can be regulated in a density-dependent manner, where if there are more individuals, then the population's growth declines, or the population's size could begin to decline. There's also density-independent factors that regulate population size or population growth, and these do not care how many individuals there are in the population. So something like competition is very clearly a density-dependent factor. All right. Predation is also a density-dependent factor. Disease is a density-dependent factor because disease pressure becomes heavier as you get a more densely packed population. Make sense? Okay. Density-independent factors are usually things like, as you see on the slide, drought, fire, habitat destruction, or some sort of random event that occurs. A stochastic event is the word that I was looking for. Something that doesn't care how many individuals there are. All right? The event that regulated population growth rate or size occurred independent of how densely packed the population was. And sometimes there will be overlap, like a species of tree that encourages fire. A more densely packed population of eucalyptus trees, there's more likely there will be fire. So now, is the fire a density-dependent factor in population regulation? You could certainly make the argument. So whenever we set up a dichotomy like this, there's always overlap. Okay. Solid. Whoa. Yeah, so if you're trying to figure out if a factor is density dependent or independent, just think about, hey, is that pressure going to become a bigger deal as the density gets higher? If it becomes a bigger pressure as the density of the population increases, it's density dependent. Cool? And vice versa. Now, R versus K selected species. Species have what we call different life history patterns. They have different adaptations for, when we're talking about a species' life history, we're talking about it's born, it reproduces, and then it dies. But there are all of these questions, right? You've got birth, okay? You've got reproduction, and then you've got, I'm just going to put death right here. And that's the population, that's an individual's sort of life cycle, okay? But there are all these questions, like how long, okay, between birth and reproduction? And then for reproduction, how often, all right? Does the organism repeat the reproductive cycle a few times in its lifespan? And then, how long does it live? Okay, how long before it dies? All right. Especially of interest can be how long does an individual survive after what we call post-reproduction, when it's no longer biologically capable of reproduction. Does it continue to persist? And if so, how long? Okay. So if that's our very basic generalized sort of life cycle, and a life history answers these questions. Describing a species' life history tells us how long is it going to live? How long does it take to become reproductively active? How many times does it reproduce? And also, how many offspring does it have? That's another big question. Okay? Number of offspring. Because some individuals, right, you think about a dandelion. Seed lands, completes its life cycle really rapidly, makes as many offspring as possible, dumps them into the environment. Okay? A dandelion's a good example of what we would call an R-selected species, meaning it has evolved to maximize its intrinsic population growth rate. And the population's growth is usually at that R portion of the curve and doesn't reach carrying capacity. All right? Yeah.
Would that number of offspring kind of like cycle through that how often thing if you had the space to draw it like that when you did it? Like would it go like up from how often and then back through that circle kind of?
It's more, I'm trying more to make it make it specific to the reproductive event, because there are species that reproduce many times, and the number of times you reproduce it's usually linked to how many offspring you make each time, but it's not necessarily linked to it. So it'd be better if that arrow was pointing the other way, I think, if anything. Does that make more sense a little bit? Okay, awesome. Cool. So, yeah, we've looked, let me not just drink water in the middle of a sentence. It's not the most professional. Okay, so look at this curve, okay? Here's sort of R max. When you're at R max, you're essentially doing exponential growth. You're growing as rapidly as possible. When you reach K, you're at the max population size. So there are two categories of life histories, the R and the K selected species. R selected species, I'll just point out a few key things here. They complete their life cycle really quickly. Short life, rapid growth, early maturity, right? They make a lot of offspring. They usually don't invest much into the offspring. They don't care for the offspring, and they don't put a lot of energy into each offspring. So you make a ton of little offspring, and you just assume that some of them are going to survive. Our selected species tend to do best in unstable environments. So they'll do really well following a disturbance. These include like the pioneer species or the primary colonizing species, okay? They do best by generalizing their niche, so they can get in there when resources are available and just rapidly complete their life cycle, produce a lot of offspring, and then perish, okay? There's a bunch of them. They tend to be prey. The population size is regulated mainly by extrinsic factors because they don't usually reach their actual carrying capacity in a given environment. They're usually, so there's two ways to think about our selected species. You can think about them as they have life history characteristics to give them a high R submax value, a high intrinsic growth rate, or you could say they've evolved to live here, all right? The population isn't really designed to ever reach carrying capacity. Okay, then we've got the K-selected species. The K-selected species have the opposite of all the things I just told you. They usually take longer to carry out their life cycle. They usually take longer to enter maturity. They usually grow slower. They usually make fewer offspring. They usually care for their offspring because what they are adapted to do is maintain the population size at carrying capacity. So they invest a lot into their offspring, and then both before birth and after birth, they put many resources into the embryo or the seed or a bunch of structures to keep the offspring inside and keep feeding it, as it can reach more mature levels. Or they make really big mondo seeds. Yes.
Case-selected species tend to be more like mammals.
Well, I'll usually use mouse and elephant. Mouse, elephant, they're both mammals, right? Yeah. So I wouldn't go that far with the generalization. Yeah, the problem is the categories are so broad, there's going to be a ton of variability within each category. Yeah.
Sorry, can I just try to rephrase what you're saying?
I would love that.
In a different way that would maybe help me understand.
Yeah, if it'll help you, it'll probably help somebody else in the room.
Is essentially what you're saying, some species try to regulate how much fluctuation they have in their, where they are around their carrying capacity more than others, or is that
No, that's great. Yeah, absolutely. So it's sort of another aspect of it, right? Like if you're a K-selected species, you don't want to have these massive boom and bust things, all right? Because if they're too big, you could go all the way down to an unsustainable population. So a K-selected species is likely to have smaller fluctuations around the carrying capacity.
maybe more likely to have bigger?
It's usually going to go up and then crash.
Okay.
Yeah, and then its offspring will move. Or if they're plants, they'll put a bunch into the seed bank and wait for the next disturbance to open up resources.
Awesome.
Very good. I love that. Thank you so much. Okay, so R versus K selected species. Big, broad categories. You're going to run into them again, I promise you, if you take any class even tangentially related to ecology or population. Okay. This is another thing. It will show up again in your classes that are about population ecology. Okay, and this is a very intuitive model, I promise. It says that a population's growth rate, is determined from a combination of birth rates, immigration rates, death rates, and emigration rates. The birth and immigration rates, that's when individuals are born into the population, or when individuals of the same species move into the population, okay? The death rate is when individuals die and leave the population, or when individuals emigrate out of that population, all right? So that's why we've got a positive term for B and I, and a negative term for D and E. Fair enough? So theoretically, if you knew these birth rates and the immigration, emigration, and death rates, you could calculate a reproductive rate out of that. All right, but this is really more useful as just a model to think about what determines a population's growth rate. If death rates increase, you get it. I'm about to just get so repetitive, this is gonna lose everybody because I'm boring myself going so in depth into this model. Reasonable enough? Okay, awesome, bless you. Good. Another thing that we use to describe a species' life history is a survivorship curve, okay? So what I've been talking about today, right? I told you, there's these models for population growth rates. And then there are different life histories of individuals where they vary in these sorts of things on the board, right? Now we've got some broad categories that we can use to describe species. And if somebody told you, it's an R-selected species, or if somebody said, that's classic K-selected species, you'd have an idea of what they meant, okay? Now we're going to also look at survivorship curves, which are also used to classify species. It's just another scheme, all right? It's another scheme. So it's focused on how long individuals tend to live, specifically. Cool. Or you could say when the death rates are higher for the individual species. Okay, cool. So check this out, right? 3 general patterns, okay? Type 1, type 2, type 3. So we have type 1, type 2, and type 3 survivorship curves on top of R versus K selected species. Whenever I have to remember this, I just go, okay, I draw the corner of a room, and then I lean a giant pistachio over here. And then I just go 1, 2, 3. And that's how I remember it. This is the giant pistachio model. All right? So check this out. Age on the x-axis, number of individuals on the y-axis. So type 1 survivorship curves. The young tend to live. You know, you get high survivor rates. These are probably K-selected species. We're taking care of the young, investing a lot of resources, not having a bunch of offspring at each event. And then they reach an age where they are usually, it's, you know, post-reproductive usually, where death rates start to go up, survivorship rates start to decline. Okay? For some individuals, they die at a pretty uniform rate throughout each age. Okay? The proportion of the population that dies during each year is pretty constant. Question?
Okay, you actually just answered my question.
Oh, awesome.
I was going to ask if the death rate was pretty constant or if it was...
Yeah. So that's, it's really easy. And I told myself I wouldn't get into the log scale, but someday you'll have to get into the log scale. Look at how each one of these units, it goes from 0.1 to 1 to 10 to 100 to 1,000, because this is all about what proportion of the population dies in each year. So you can see right here, this goes from 100 to 1,000 during that time, and then it goes from 100 to 10 in that same period of time. But the proportion of the population that died was the same. 90% of the population died from each time point. All right. If you don't log transform this data, it doesn't account for the fact that you're trying to look at a proportion of the population. Cool? Solid. Solid. Log transformation is important. Blah, 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 blah. So check it out. Type 3. We've got the type 3 survivorship curve, the bottom of the pistachio, right there. Usually R-selected species. Make a ton of offspring. Many of them perish, but a few survive. All right? So the survivorship curve, so the survivors of that initial high death rate do a little better. All right? Most of the individuals die at a young age. Once they've gotten established, they can be okay. Cool. So type 1, type 3, those are the extremes. Type 2 covers some of the intermediate individuals with intermediate survivorship. And these are survivorship curves. And they tell us how long species survive. Okay. Population regulation. We're going to focus a little on the density dependent and density independent. Just the density dependent is what it looks like what I chose to go with right here. Very good. Okay? This is good because it does the classic lynx and snowshoe hare graph where both populations are, both populations are put on the same timeline. Pretty solid. So there are both interspecific interactions and intraspecific interactions which become a stronger pressure as densities get higher. So of course things like intraspecific competition, okay? That becomes more of a pressure as the density gets to higher levels. Disease, okay? Now, if the disease is due to a pathogen or it's vectored by a symbiote, is that an interspecific interaction? Not a symbiote. Yeah, well, it's symbiote. Yeah, it works. Cool. So we've got interspecific interactions between species, density-dependent regulation of a population, predator-prey oscillations. Both the predator and the prey regulate, to some extent, the population size of the other. So the prey population goes up, that lets the predator population climb. The predator population gets too high, it causes the prey population to dip. So then the predator population dips, and then the prey population goes up. This is a classic predator-prey oscillation, And if you think there's not another model for this, then you haven't been paying attention. But we're not going to worry too much about it. It's just the classic ones, the Lotka-Volterra predator-prey model. There are 2 R's in Volterra? Yeah, there are. Let me just show you real quick. Oh, I should have put equation. There's 2 equations, multiple equations. Here we go. This is a good one right here. Open the image in a new tab. Boom. So when you're modeling multiple species and how they're going to be influencing one another's growth rates, you're essentially just using the logistic model, right? Except you're including the carrying capacity for that species as well as the effect of the other species on that species. This looks intimidating, and so we're not going to dwell on it for too long, but when you hit this in your future classes, just know that it's a slight modification of the logistic growth model that has to include some term for the effect of each species, the predator on the prey and the prey on the predator. Okay? Cool. This is the competition model, not the predator-prey model, but they're essentially the same set of equations. Okay, cool. Cool. So that's also dependent on density, solid. I think we've kind of covered it. I'm feeling pretty good about that. Good. Minimum viable population sizes. So in conservation, it's usually important for us to know the size of, at what point a population is no longer viable. So there's what we call the minimum viable population size, the MVP, okay? For a given species, for a given population, a species within a certain habitat. So yeah, it is determined usually by some sort of a computer model, all right, and historical records for species that have been there, for populations that have been there in the past. If you've been doing a lot of survey, you can get a good idea of what the minimum viable population is, but you need to know the minimum viable population terminology, okay? All right, maybe it's an endangered species, maybe there is going to be an impact that you can't stop happening in an area and you know it's going to reduce the population of the species by 40%. And you want to know, is that population reduction going to reduce the population size below the minimum viable population? I don't want that term to, be a surprising one for you. Okay? Cool. Island biogeography. Island biogeography is very important for developing these models, okay? Very important for developing these models, and it's where a lot of our generalizations about what makes a population viable sort of come from, okay? So remember, a population's growth rate isn't just determined by birth and death, it's also determined by how many individuals of that species are coming into the population and how many individuals are leaving that population, okay? So populations aren't usually entirely isolated and distinct. There's usually a bunch of spatially separated subpopulations that have some genetic flow between them, some flow of individuals. Or if it's plants, it might be pollen, all right, is flowing between these different subpopulations. So it's all about the movement of genetics between these subpopulations. If there's genetic movement between individuals, the populations are linked. Fair enough? Solid. So we call that a metapopulation. A metapopulation is a group of small populations linked by gene flow, either through mature individuals or through propagules, seeds, pollen, etc. Good. Stupendous. So the theories for metapopulations and what makes them stable if you look at this, it looks just like a little group of islands floating out in the ocean, right? So researchers investigated all these different island chains, and they looked at how important proximity to other populations were, as well as size of the individual islands. So talking about viability of populations, island biogeography produced simplified models that focus on how easily can you get new individuals to move in, so proximity to another population, and the size of the habitat. So in island biogeography, it's size of the island and how close you are to the mainland or to a larger island that's a source population. It leads to terminology like source populations and sink populations, which are exactly what they sound like. A source population produces excess individuals, which then emigrate out, and a sink population, it produces it does not produce enough individuals to lead to a sustainable population, but it is supplemented by individuals moving in or immigrating into that population. All right? So this is the metapopulation concept. This is the big findings come from island biogeography and metapopulations. Pretty cool stuff. Friction. Okay? Friction's a term, and once again, I'm going to do this again. Like, every textbook talks about metapopulations, but not every textbook talks about friction. Okay? And friction's a really important thing to think about. It's how much resistance there is to movement between the subpopulations. So that friction, value is usually represented as a decimal that modifies immigration or emigration rates between populations. And it's all modeled and it's used to predict, hey, are enough individuals going to come in to keep that population's growth rate positive? Reasonable enough? Okay. Island biogeography. Just what I was saying before, okay? How far are you from the mainland? How far are you from a source population? Can individuals make it? Is all these concepts about, if a population undergoes local extinction, can it be replenished? Can it be recolonized? All right. Source sink dynamics we talked about. Very good. I love it. Complementation is a concept where the different subpopulations might have different resource types, and so it helps the species complete its life cycles. Like maybe a population exists in one habitat type moves between different habitat types during different stages of its life cycle because that has resources that it needs. So you don't have to know all these insights from island biogeography, but island biogeography got a fancy name. It's not that complicated of a concept. And you just read Future of Life, and E.O. Wilson's one of the big, founding people behind island biogeography. So him and some other researchers fumigated small islands and then looked at how quickly the insect populations replenished on those islands. That's where you get a lot of these findings for metapopulation dynamics. Okay, we're almost there. We're doing pretty well. I got 10 minutes, no problem. No problem. Conservation. Classic conservation, number of individuals. What do? We maintain the number of individuals. That's what we can do. We can count the number of individuals. Modern technology. Let's just look at the genetics. What we're really trying to do in conservation, at least from the view of the biologist, is you're trying to maintain genetic variability, right? You want that species worldwide to have a lot of genetic variability. And if you really sit and think about it, different species are just things that have more genetic variability. So your goal as a conservationist trying to maintain species diversity is still maintaining a lot of genetic diversity on the entire globe. All right? maybe that works for you, maybe it doesn't. It's fine either way. Genetic diversity. We've got better techniques now to identify individuals that have greater genetic diversity or less genetic diversity. If an immigration source gets cut off from a subpopulation, you can look at that and say, oh, they're not going to be receiving more genetic diversity. Maybe we need to do a translocation of individuals between these two subpopulations. Genetic diversity, conservation genetics, conserving genetic diversity. Are the big hand movements working? And take, put them here. Good. All right. So we're going to talk a little bit about things that affect genetic diversity. Okay? We've spoken already about evolution and all the different factors that can change the allele frequencies of a population, okay, from generation to generation. And during that same time, I talked to you about genetic drift, and I said, we'll talk more about genetic drift later. And here, as promised, more genetic drift. So genetic drift is random, okay, changes to the gene pool, all right? Or it's a situation where you get a random genetic diversity in a population, okay? It's not necessarily better adapted to its environment. It's just a random suite of genes that you've got. So this includes things like the founder effect, okay? And in the founder effect, there's a few individuals that start a population, so you are limited in genetic diversity to the individuals that founded the population. Reasonable enough? In a bottleneck effect, all right, a bunch of individuals get removed, a drastic reduction in population occurs, okay? And you're left with whatever genes survived that massive culling event, okay? So these sort of lay, how do I write it here? They lay the background. They lay like the background for the genetic diversity of the population. And then natural selection can act on the genetics that are there. Evolution can occur within that population then. But these are sort of drastic shifts that are due to chance. They're random. Okay? Cool? If you reduce your genetic diversity, you can get what's called inbreeding depression. Really, people summarize inbreeding depression by saying you're more likely to accumulate deleterious alleles. Inbreeding's complicated, okay? What actually leads to the negative impacts is complicated. But there's a generally recognized phenomenon of if a population has very reduced genetic diversity and it continues to breed within that population, you get sort of a spiral where the individuals start to feel worse, perform worse. Yeah?
I haven't really looked into it, but I've heard of, like, for example, like people who breed snakes are able to breed like the mother with the son.
Yeah.
How does it not be an issue?
Absolutely. So it's a highly managed sort of breeding situation, right? And what you do is you repeat, it's dark, but you repeatedly cull individuals that make unfit offspring. So then you're like, okay, well, these bred and they made offspring that didn't work, so they're out of the gene pool. And now let's breed this mother with its offspring and then see how those individuals work. Because one of the reasons that you tend to get inbreeding depression is because of masked recessive alleles. So I don't know if you remember when we talked about heterozygotes, right? And usually when you're homozygous recessive, not usually, many times when you have two copies of the recessive allele, you don't make the protein. So you have individuals that are heterozygous and they're prevalent in the population and they're breeding, and 1 out of 4 times, or 1 out of 4 times, right? They make a homozygous recessive offspring. And then that offspring persists, it survives to breed again, and it slowly increases the prevalence of the recessive alleles. It's not hyper-intuitive, but that's the pattern that's noticed. So if you're selecting against heterozygotes and only getting the dominant or desired forms of the alleles, then you can avoid inbreeding depression. But boy, oh boy, that's an intensely managed system. Yeah.
It seems like there's a lot of like
Absolutely. Yeah, totally. Especially when it's in the natural system, you know. Good question. I love it. So yeah, I mean, seriously, like looking into inbreeding depression is interesting. There are other reasons that inbreeding tends to lead to less fit offspring. Cool. Population viability analyses and minimum viable populations. Okay, so I already talked to you about minimum viable population, what that number means. Put that screen back down because it's driving me nuts just cutting it off at the bottom there. The minimum viable population is the population size where researchers predict it will it'll be able to maintain. All right, there's enough individuals to maintain the population size. There's enough individuals to prevent a crash due to inbreeding depression. It's different for different species, all right? And you determine the minimum viable population using a population viability analysis, which is basically a computer model. Well, it's a model for each species. You plug in a bunch of variables about the population size, and if you know it, the genetic diversity, and then that thing just models the population's growth and throws random stochastic events that shove the population size down on it. And then it predicts, oh, can the population maintain? Because those stochastic disturbance events that reduce the population size and maybe genetic diversity are going to happen. The question is really, is the population size large enough to handle those genetic drift events? All right, question.
So when it's below the minimum viable population, would that be when like humans would typically intervene to kind of prevent it from going extinct?
Yes, that's the idea behind this, right? And so the way that you look at the minimum viable population size is usually you run that model with you run it 10,000 times, all right? And then the minimum viable population size is the population size where I think 95% of those runs lead to the population being stable for 10,000 years. There's numbers associated with all of it that are just, somebody has determined that that was good, and then everybody's stuck with it. All right? So, yeah, we're not going to you're not going to run a population viability analysis here. I just want you to know the term minimum viable population size. It's, once again, sort of what it sounds like. But don't forget, it's also incorporating, can it handle random stochastic events? And does it have enough genetic diversity? So I've got some slides from a previous lecture that talked about community dynamics. And look, I even put myself a note. Use them as a refresher if we have extra time, which we don't have a ton of extra time. But remember this stuff: species richness, dominance, evenness, diversity, how we measure it all, right? How we measure diversity, the different equations. Cool. There are a few things I want you to leave ESM 105 with, and one of them is I want you to have a more nuanced understanding of what ecologists mean when they say diversity to you. Okay? Solid, just so you can chat with them. Structures, connectedness, diverse, complex, primary productivity. We talked about all this stuff previously. Great. Well, there we are. It's 9:49. We got through the week 6 PowerPoint on Monday of week 7. I would say review those last slides if you get a chance. We're going to jump into week 7, 8 PowerPoint. It's all about biodiversity. It's going to be a lot of fun. There's a couple of unexpected soapbox moments that I'm very excited to get on with you. Yeah. And then week 9 is set aside for catch-up. So if we go slow through this, we're in no rush. Ask lots of questions. I'm holding on to you. Get out of here. Have a great day. Have a great day. Farewell. Goodbye. Thank you.
Thank you.
Of course, thank you.